Hello, everyone. We are from the Department Biological Safety team, bringing to you a safety training video on biological spill cleanup in a centrifuge. During centrifugation, spills or breakages can happen, and this poses a serious potential for exposure to aerosols. Aerosols are formed when fluid escapes from the rotor or cups while centrifuging at high speed. Aerosols is one of the main causes of lab-acquired infection. Spills could occur from either inside the bucket or could leak from the broken bucket. If a centrifuge tube breaks when the centrifuge is running, turn off motor immediately. Leave the centrifuge lid closed and evacuate room. If breakage is discovered after the machine has stopped, reclose the lid immediately. Alert and inform staff in the immediate vicinity of the spill to evacuate area using normal exit procedures. Barricade the centrifuge and vicinity around it. Post a warning sign on the centrifuge and the laboratory door informing that there is a biohazard spill and indicating the time of spill. Allow aerosols to settle for 30 minutes before initiating the spill cleanup. Do not allow others to come near the spill area. Notify your supervisor and safety officer. Steps in donning PPE for biological spill cleanup. First, inner gloves. Second, N95 mask. Third, safety goggles. Fourth, disposable lab coat. Fifth, hairnet. Sixth, outer gloves. Make sure the gloves are over the cuffs of the gown. And lastly, shoe covers. Contents of biological spill kit. Disinfectant, freshly prepared 10,000 ppm chlorine. Waste container, biohazard bags, sharps bin. PPE, disposable lab coat, gloves, safety goggles, N95 mask. Mechanical tools, dustpan, broom, tongs. Accessories, tapes, signages. Have a complete biological spill kit ready to go before you start the cleanup. Assess the extent of contaminated area. Consult the SOP and risk assessment to identify a decontaminating solution that will be both effective against the offending pathogen and that will not compromise aspects of the centrifuge itself. Ensure risk of injury is controlled prior to initiating spill cleanup. Decontaminate and remove all items within spill area. Remove rotors and sealed buckets to the nearest BSC before opening. Use tongs or forceps to remove any broken pieces of tubes and dispose them in a sharps container. Do not pour decontaminant directly onto a spill as this can produce more aerosols. Use a pre-soaked paper towel to decontaminate that area. Allow 30 minutes contact time to ensure germicidal action of disinfectant. Dispose all contaminated paper towels in the biohazard waste container. Reapply disinfectant to the affected area and after 30 minutes contact time, wipe up the area. Repeat if necessary. Rinse with water to prevent corrosion to the centrifuge. Biological spill cleanup in a centrifuge bucket. Carefully remove any unbroken tubes of samples and place them into a biohazard bin. Be careful not to spill the contents. Thoroughly disinfect and soak the inside and outside of the centrifuge rotor, cups, and accessories and allow 30 minutes contact time. After disinfection, move to the sink to rinse and dry thoroughly. Disinfect the rotor using appropriate disinfectant. Check the equipment product sheet for correct way to disinfect. Place autoclavable contaminated reusable items in biohazard bags or heat-resistant pans or containers with lids before autoclaving and further cleanup. After disinfection, remove PPE in the correct order. First, shoe covers. Second, gloves outer layer. Third, disposable lab gown inside out. 4. Safety goggles. Fifth, N95 mask. 6. Hairnet. 7. Gloves inner layer. Dispose all cleanup materials in a double biohazard waste bag and label the bag with date and PI lab. Report the incident to ORMC via the NUS Accident and Incident Management System, AIMS. Lastly, replenish the biological spill kit contents. Here, introducing our biological spill responders. We have come to the end of the training session. Thank you for your attention. We hope you have benefited from this training video. Goodbye.